Hello students, in this video lecture we are going to learn about the transcriptional modification. After the transcription, the mRNA, premature mRNA, it will be undergo some modifications. So those modifications are called post transcriptional modifications, right? So first we will see the what is the exact process which is going on in the post transcriptional modification. So the nascent RNA, that is the premature mRNA, are also known as a primary transcript. So it needs to undergo some modifications to become the functional or mature tRNA, rRNA and mRNA, right? So primary transcripts of mRNA are called as a heterogeneous nuclear RNA, that is hnRNA, right? So these hnRNAs are larger than the matured mRNA because you know the eukaryotic genes, right? So they have exon as well as intron. So after the transcription they have exon and introns region. So exon region have to be joined and introns region have to be removed. So that's why after the removal of the introns region, only exon region will be there in the matured mRNA. That's why hnRNAs are larger than matured mRNA, right? So these are the post transcriptional modification, like the splicing, addition of 5 prime cap, creation of poly A tail and RNA editing, right? So first we'll see the splicing. So splicing in the most of the eukaryotic genes, okay, compared to the typical prokaryotic genes, are interrupted by the non-coding DNA, right? So during the transcription, the RNA polymerase transcribe the entire gene without any distinguishing between the coding and non-coding regions. So that's why the coding regions it refers to as a exon, while non-coding region are called as a introns, right? So only exons are responsible for the protein synthesis, right? So such kind of structural genes are composed of coding and non-coding region, right? So these non-coding sequences called the interventing sequences of or introns, or coding sequences are exons, right? So these have to be get removed. So here we will see the mechanism behind this. The splicing signals in the nuclear mRNA, and that is the precursors, they are uniform, right? So you know the spliceosome. So these spliceosomes, they act on at the nucleolus, right? So these splicing signals in the nuclear mRNA, they act as a precursor, and first two bases of the introns are almost always with the guanine and uracil, and at the last two are adenine and guanine. So in addition to these two sequences at the intron exon boundaries, another sequence that is called as a branch point. Okay, so you can see here branch point also. So this branch point sequence upstream to the 3 prime end of the introns is very important, right? Because so these are having the OH group, and that oxygen is having the lone pair of electron, and it will be it will cause the nucleophilic attack at the exon end, that is GU end, what you can see here, right? So the splicing reaction is catalyzed by the spliceosomes, right, which composed of many proteins as well as smaller nuclear ribosomal ribonuclear proteins, that is snRNPs. So in this figure the mechanism is shown of splicing in a pre-mRNAs, right? So the typical mammalian pre-mRNAs has many introns, so therefore this splicing process is must ensure that the exons are spliced in the correct order, right? So every precursor mRNA has the unique pathway for the removal of introns during the splicing. So dictate that by the conformation of RNA that influence the accessibility of the splice site. So the 3 prime and the 5 prime and 3 prime splice site, and the branch sequences are recognized by the components of splicing apparatus, so that assembles to form the large complex that is called as a spliceosome. Excuse me. So the snRNPs they involved in the splicing are U1, U2, right? U4, U5 and U6. So each snRNPs they contains a single small nuclear RNAs, that is called as a snRNA, and several proteins. So the U4 and U6, okay, the snRNPs are usually found in the single particles, means common structural core proteins are found to be associated with all these snRNPs. So in addition, each snRNP has a unique protein associated with it. The core proteins, which are called as a ESM proteins, and they bind to the conjugate sequence in the snRNA. So in addition to these proteins associated with the snRNA, there are 70 other proteins found in this spliceosome, that is called as the splicing factors, right? So this protein includes the required which are required for the assembly of the spliceosomes, and the proteins which are involved in the catalytic processes, right? So therefore there are two distinct types of splicing reaction. So first we'll start with the consensus sequence at the 5' splice site, the branching sequence and the adjacent pyrimidine tracks are recognized when the complex assembles, right? So first complex formed during the splicing reaction is complex E, right? So this complex E, which contains the UN snRNPs, that is the splicing factors, then U2AF and the members of the family, that is called as a SR proteins, which are splicing factors and regulators, okay? So sorry, so you can see the introns, right, which is start with the GU sequence and end at the AG sequence, and there is a branch site, that is denoted by A, adenine, okay? So U2 will be bind at the branch site and the U1 will be bind at the GU site, okay, starting point of introns, right? Then after that U4, U5, U6 complex will be get together and they will attack on that, okay? And after that, after the utilization of ATP dependent protein mediated unwinding will be takes place, and the lyrate will be formed, and in this way the introns will be get removed, right? But there is a very huge mechanism behind this. So the E complex is also called as the commitment complex, since it identified the pre-mRNA as a substrate for the formation of splicing complex. So in the E complex, the U2AF, okay, so U2AF, yes, you can see in this PPT the this complex at the branch site, okay, these proteins will be bind first, okay, and another splicing factor, that is the SF1, also bind to the E complex, and the connection in between the U2 and the U1 bound at the 5' splice site. So the E complex is converted to the A complex when the U2 snRNP bind to the branching site. You can see the branching site here, okay? So both the U1 and U1 snRNP, U2AF are needed for the U2 binding, otherwise U2 cannot bind. So U2 snRNAs has a sequence complementary to the branch sequences. The binding of the U2 snRNP requires the ATP hydrolysis and commit the branch commit the pre-mRNA to the splicing pathway. So which following the complex E, the complex B1 is formed when the trimers containing the U5, U4 and U6 snRP will going to bind. So this complex is regarded as a spliceosomes because they will cleave this. So that's why called as spliceosomes, right? Then after that the dissociation of U1 is required for other components to come into the juxta position with the 5' splice position, notably the U6, right? So U6 will be bind only when the U1 will going to remove, okay? So at this point the U5 snRNA which changes its position, the initially it close to the exon sequence at the 5' plus site, but in the B2 complex it shifted close to the introns sequences. Okay, so here you can see the formation of this complex. Okay, the production of the antibodies against the small nuclear ribosome causes the systemic lupus erythematosus. So if the debate will be take place. Okay, so you can see here the E complex with the UN U2 with the SF2, it bound to the branching site. Then B1 complex will form with the trimers binding at the U5 and at the 5 prime, and U6 bind to the U2. Okay, then this B2 B1 complex will convert into the B2 complex with the U1, it will be get released. Okay, uh, U5 shifts and U6 bind at the 3 prime place site, and then C1 complex will form by the U4 release and the 5 prime splice site cleaved at the lyrate form. So here you can see the lyrate will be get removed, right? So this C2 complex at the 3 prime splice site cleaved and the exon ligated and the spliced RNase is released and lyrate is debranched. Okay, then we will see the next modification, that is, so here you can see the two exons, they get bind to each other as their end, they are they attract to each other, and thus they attach and forms the mature mRNA. Okay, then next modification, that is the 5 prime capping. So most eukaryotic excuse me, so most eukaryotic mRNAs have a 5 prime cap, right? So yes, a residue of 7 methyl guanosine linked to the 5 prime terminal residue of the mRNA through an unusual 5 prime triphosphate linkage. Okay, so here what will happen, the formation of cap is required in the 3 steps. In the first step, what will happen, at the 5 prime terminal phosphate groups, one phosphate group will be removed by the phosphatase enzymes which leaving the diphosphate, means at the 5 prime end there are 3 phosphate group, one phosphate group will be get removed by the phosphatase enzyme. You know what is the function of phosphatase, it cleaves the phosphate group, it removes the phosphate group. So how many phosphate group will be there, only 2, because one is get removed. So this is the first step of 5 prime capping, and at the second stage the GTP is added by the excuse me, by releasing the pyrophosphate. Okay, and at the third step, the seventh n of the n means any nucleotide sequence of the guanine is methylated by the methyl transferase enzyme. So methyl group donor is a S adenosyl methionine. Okay, so this will going to donate the methyl group. Then at the end the poly A tail will going to form at the 3 prime end. Okay, so why there is a need of poly A tail addition, because it provides the stability to the matured mRNA, and it also provides the protection to the matured mRNA from the exonuclease. So exonuclease is there spread in the cytoplasm. So after the transcription, after the formation of RNA, so RNA will be come out in the cytoplasm from the nucleus in the eukaryotic cells, and thus in the cytoplasm there is an enzyme like the exonuclease enzyme, so it can attack at the 3 prime end of the RNAs. So that's why these RNAs have to be get protected, and the protection will be provided by the addition of poly A tail. Poly means many, A means adenine. Okay, and tail, why we will call it as a tail, because at the tail region, at the end region, at the 3 prime end region, this continuous adenine will going to form, that's why it is called as a poly A tail. Okay, so at their 3 prime end, most eukaryotic mRNA have a string of 40 to 200 adenine residues, which making up the poly A tail. So adenine nucleotides are added by the enzymes, that is the adenylate transferase enzyme. So this adenylate transferase enzyme will add continuous adenine, adenine, adenine at the 3' end. Okay, so here you can see the RNA editing also. So central dogma states that the DNA it's transcribed into a sequence of RNA that is in turn directly translated into the protein. RNA splicing adds an additional step into the processing of the gene expression, wherein the interrupted sequences get removed and the coding sequences are joined. So this coding sequence in the DNA they remains unchanged. Alright. So changes in the information coded in the DNA occurs in few cases that leads to the generation of new coding sequences. So which means the information changes at the level of mRNA. Okay. So therefore coding sequence in the RNA differs from the sequence of DNA from which it going to transcribe. So this process is known as a RNA editing. Okay. So during RNA editing, the bases can be added, deleted, or substituted. Okay. An example of RNA editing where the basis is substituted is apolipoproteins gene of mRNA. Okay. So this RNA is translated into the protein representing the full coding sequences in the human liver, right? Whereas the shoulder protein is synthesized in the intestine. So this protein is translated from the mRNA whose sequence is identical to that of the liver except for the change from cytosine to uracil of codon. So this substitution changes the codon. Codon means you know the triplet codon. Okay. So you can see the in the intestine, so there will be addition of the substitution of the codon. Okay, and such process is called as a RNA editing. So there is a differences in the liver and intestine. Okay, so you can see this substitution changes the codon. Okay, so thus the editing event in the ApoB mRNA causes the single cytosine at this specific position to uridine and thereby produce a smaller proteins. So this substitution is achieved by the deamination, whereby an amino group of the nucleotides ring is removed and it is catalyzed by an enzyme that is called as the cytidine deaminase. So this cytidine deaminase has a catalytic subunit which is released in the bacterial cytidine deaminase, but has an additional sequence recognition domain. So that helps to recognize the specific site for the editing. Okay, so in this way the editing takes place. Modification of tRNA, the primary transcripts of the prokaryotic and eukaryotic tRNAs, they are processed by the removal of sequences from each end, that is the cleavage end, and in the few cases by the removal of the introns, that is splicing. Okay, so you can see here the basis modification at the 3' cleavage or the 3' or 5' cleavage, or cytosine adenine addition. So this codon is also get modified here, and thus the mature tRNA get formed. Okay, so here you can see the base modification here, the methylation at the adenine to the guanine. Okay, or the reduction of uridine into the DHU, or the transversion of uridine into the sigma factor, or the deamination of adenine to the first guanosine. Okay, so this base modification can be takes place here. You can see the methylation as well as the nuclease which cleaves this kind of group. Okay, and thus the processing of these eukaryotic RNAs will be takes place. So there is a continuous sequences, but some have get removed, and thus 18S, 5.8S and 28S subunit get formed, and they will get together to form the ribosomes in the eukaryotic system. What are the inhibitors of transcription? The antibiotic like the actinomycin D, it binds with the DNA template and block the movement of RNA polymerase. Then the rifampicin, this antibiotic used to treat the tuberculosis, that is TB. So it binds with the beta subunit of prokaryotic RNA polymerase. Okay. Then another antibiotic, that is the amanitin. So this toxin produces the Amanita phylloids mushroom. So this Amanita phylloid mushroom, mm, it, it it secretes such kind of toxin. So it tightly binds with the eukaryotic RNA polymerase second, and thus it inhibits the mRNA synthesis in the eukaryotic system. So these are some of the inhibitors of transcription process. And there are the retrovirus like the cold virus also, they possess the RNA as a genetic material, and thus when they attack on their host, okay, then they have the enzyme, that is the RNA dependent DNA polymerase. So this DNA, they are the complementary to the viral RNAs. Okay, the cDNA, it can be used as a probe to identify the sequence of this DNA in a genes. So these retroviruses, they perform the reverse transcription, means their genetic material is RNA, but when they are affecting the host, when they are attack on the host cell, then what will happen, they will inject their RNA inside the host cell, but host having DNA as a genetic material, right? So that's why what they have the machinery, means they have the enzyme that is called the RNA dependent DNA polymerase. Okay, so what happen, the RNA will further convert into the DNA. That process is called the reverse transcription. So they perform reverse transcription, okay, and they use the host machinery to convert that DNA into the RNA. Okay, it can be replicated also, it can be transcribed also, and it can be translated also. After the translation, the protein will synthesize, their body structure will form, machinery will form, and thus they complete their life cycle. So here you can see this RNA genome, they insert the RNA, the reverse transcription takes place. Okay, and inside the nucleus, their genetic material get stuck with the genetic material of the host cell, and in this way they perform their life cycle. Okay, so we will stop here. Thank you.